Hello, everyone, and welcome to this AnaBios webinar. My name is Chris Mathis, and I'm the Chief Commercial Officer at AnaBios. It's my pleasure to be the moderator for today's session. So AnaBios has been hosting webinars on various preclinical drug discovery topics over the past several years, and it's our pleasure to present today's webinar, which is another in our long series of webinars on the topic of pain research. So many of you know AnaBios already, but for those that don't, I'll tell you a little bit about us. So AnaBios is a unique contract research organization and biotech company based in San Diego, California. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human, and working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. So before I introduce our speaker today, I just want to mention a couple of things. If you look at your GoToWebinar panel, there's a questions tab. So you can enter questions throughout the presentation, and we will get to those at the end. I think we'll have plenty of time for that. Also, you can raise your hand, and I will call on you, and you can ask the question verbally. Also, you might notice in that panel there are some handouts we have available for you to download: the AnaBios Human DRG brochure and the AnaBios Spinal Cord brochure. So those are available throughout the presentation. All right, so without further ado, I'd like to introduce our speaker today. Dr. Menichella received her MD and PhD from the University of Milan in Italy. She was a postdoctoral fellow at Wayne State University and Harvard Medical School. She graduated from the Neurology residency program at Northwestern University in Chicago, and she is an associate professor of neurology and pharmacology at Northwestern University. She is the director of the Peripheral Neuropathy Multidisciplinary Clinic and director of the Charcot-Marie Tooth Association Center for Excellence. She's actively engaged in basic science and translational research, including NIH NeuroNext clinical trials. Dr. Menichella is a principal investigator of an NIH-funded laboratory that investigates the molecular and cellular mechanisms underlying neuropathic pain and axonal degeneration in hereditary and acquired peripheral neuropathies, with a particular focus on painful diabetic neuropathy. Towards designing more effective therapeutics, Dr. Menichella's laboratory takes advantage of an integrated approach combining pain behavioral tests, electrophysiological studies, in vitro and in vivo calcium imaging studies, confocal studies, chemogenetics, and single-cell RNA sequencing with conditional and transgenic mouse models. More recently, her laboratory has been validating therapeutic targets using human tissue, including DRG and skin biopsy. So it sounds like a very well-rounded laboratory, and we are very excited to hear more. So Dr. Menichella, thanks for joining us today, and it's all yours. Please take it away.
Thank you, Chris, for the kind introduction, and also I would like to thank Gary and all the AnaBios team for the invitation to present the data from my laboratory. My laboratory investigates the molecular and cellular mechanisms underlying painful diabetic neuropathy. Today, I will present mostly unpublished data describing how we're using single-cell RNA sequencing to discover novel therapeutic targets for painful diabetic neuropathy. I have no disclosure. It is important to study painful diabetic neuropathy because diabetes is a very prevalent condition around the globe. And the World Health Organization estimates that 422 million of adults have diabetes. And diabetes is on the rise, and the percentage of population living with diabetes is predicted to increase. Painful diabetic neuropathy, PDN, is one of the most common and intractable complications of diabetes. Despite this prevalence and impact, current pharmacologically recommended first-line therapeutics for PDN are only partially effective and also have many side effects. Clinical trials have failed to show beneficial effect to treat our patient in a clinical setting. And at this time, when we are dealing with the opioid crisis, there is a pressing need to develop novel, non-opioid alternatives. PDN, it is a complex disease, but to simplify, we can say that PDN is characterized by two main hallmarks. One is neuropathic pain, which is a chronic complex pain state that often described by the patient as burning or pins and needle sensation, and this is very debilitating. The second hallmark, it is small fiber neuropathy, which is the axonal degeneration of the nerve terminal, which are the axon of the dorsal ganglion neuron carrying pain, the nociceptor axon that extend to innervate the skin and degenerate in PDN. One of the clinical barriers for the development of new effective treatment for PDN is the fact that molecular mechanism leading to both neuropathic pain and small fiber neuropathy is widely unknown. To study this mechanism, we use the well-established high-fat diet mouse model of PDN, where mice are fed either a regular diet or a diet with a high content of fat, high-fat diet, for several weeks, after which they develop obesity, glucose intolerance. These mice also develop a kind of pain hypersensitivity called mechanical allodynia and small fiber degeneration. Using this model, we have investigated the role of increased excitability and increased intracellular concentration in DRG nociceptor in the development of PDN. Now we know that DRG are heterogeneous in nature. So neuron involved in sensation of other modalities such as proprioception, mechanoreception are also housed in the DRG together with nociceptor. So if we want to study the changes within the nociceptor neuron PDN, we had to highlight them and label, and we genetically labeled them, the nociceptor population, using as molecular marker the sodium channel NAV 1.8 that labels about 90% of the nociceptor population. We did that with a genetic approach, and we crossed the NAV 1.8 Cree mouse from John Wood with the TD tomato mice, and in the mouse result, all the nociceptor are genetically labeled in red in their cell body. Here we can see the DRG nociceptor cell body in the DRG. Also, we are also able to visualize in red the axon of this DRG nociceptor extending to innervate both the dermis and the epidermis. Here we can see nicely in this confocal macrograph of skin taken from control mice fed the regular diet. On the contrary, mice fed high-fat diet, we see a large depletion of this axon, with generation of this axon. And so that demonstrates that this model developed not only mechanical allodynia, but also small fiber degeneration. So this is a good model to study PDN because recapitulate all the hallmark of the human disease. So using this model, we already shown that neuron within the DRG become hyperexcitable. And we've seen that this hyperexcitability leads to mechanical allodynia, also small fiber degeneration, which is at the loss of these intraepidermal fibers. Our hypothesis is that in PDN, DRG nociceptor become hyperexcitable, and this lead to pain hypersensitivity, but also to an increase of the intracellular concentration of calcium in nociceptor. Accumulation of toxic concentration of calcium may ultimately induce axonal degeneration, PDN. And in fact, Nirupa in our laboratory went ahead and demonstrated that blocking this excitability with a chemogenetic approach were able to prevent and also reverse not only neuropathic pain, but also axonal degeneration in this model. We are further investigate this molecular cascade and show that one way increased calcium in nociceptor leads to small fiber neuropathy is through abnormal mitochondria function. In particular, DRG hyperexcitability is associated with the increase of intracellular concentration of calcium in the DRG nociceptor that lead to excessive calcium entry into the mitochondria through a particular calcium channel, the main pathway of calcium entry into the mitochondria, called mitochondria calcium importer. So excessive calcium entry into the mitochondria lead to increased fission, therefore a fragmented morphology of mitochondria, axonal degeneration, mechanical allodynia. So now we already had published this data, but overall, those studies provided us with a better understanding as how change in excitability, calcium influx, and altered mitochondria function contribute to neuropathic pain as small fiber neuropathy. This also allowed to identify unique molecular target for the development treatment for PDN. But today, I would like to tell you about how in our laboratory, now we're taking advantage of molecular approaches that are potentially helping us to accelerate the discovery process. In particular, Dr. Dale George, a talented postdoctoral fellow in my laboratory, started to identify the complete molecular profile of DRG neuron in their hyperexcitable state in alpha-die mouse model of PDN. We wanted to use an unbiased approach that could provide multiple interesting targets that could be translated then in new, more effective, and disease-modifying therapy for our patient with PDN. Therefore, using the alpha-die mouse model of PDN, we wanted to do a transcriptomic comparison between the gene expression of the DRG neuron from mice fed alpha-die compared to mice fed a regular diet. While bulk RNA sequencing will be allowed us to see different differentially expressing genes, now the heterogeneous would be lost. In fact, the DRG neurons are molecularly defined into distinct subtypes based on the expression of a set of molecular markers. So in order to capture the full heterogeneity of the DRG neuron, a characterized change in cell types and in cell state, we perform single-cell RNA sequencing. We prepare a viable single-cell suspension from lumbar DRG from mice either fed a regular diet or an alpha diet, and we use the 10X platform for single-cell capturing and barcoding. And then we perform comparative clustering, and then we can see that we were able, with this comparative cluster, we were able to identify cell types as well as conserve markers in both conditions. We were able, as expected, we were able also to separate, identify neuronal population and non-neuronal cluster. Within the neuronal cluster, we were able to identify distinct molecular subtypes as described already in the literature. Now, for our comparative analysis, we saw that there were not large fold changes difference between transcript in the two conditions. And this is in line with what you already find in our RNA bulk RNA sequences as well. But obviously there was a wealth of information indicating several differential expressing genes and enriched pathways, including a strong indication that pointed to translational differences. Now there were very many interesting targets, but we focus on the mass-related G protein coupled receptor D. And that was also one of our hits in our bulk RNA sequencing. Now, MRGPRD, it is an interesting candidate because, as the name suggests, it is a G protein coupled receptor, make it a very druggable target. It is the excitatory receptor with a constitutive activity. So this constitutive activity of the receptor make it an ideal candidate for generating spontaneous electrical impulses as what we commonly observe in PDN. In addition, we know that the expression of this receptor can influence the excitability of the DRG neuron in response to mechanical stimuli. Also, we know that fibers that express MRGPRD extend all the way to the stratogranulosomes. This is the outermost layer of the epidermis. So those termina expressing excitatory MRGPRD receptor are a very accessible target. Now, the feature plot of our single-cell RNA sequence here shows that the expression of MRGPRD is indeed restricted to a subpopulation of neurons. Typically, neurons that express MRGPRD are classified as non-peptidergic type 1 MP1 neurons. But for our comparative clustering, we were able to identify two clusters that express MRGPRD. Here we refer them as MP1 type 1 and MP1 type 2. When we look at regular dye and high-fat dye DRG separately, we saw that while there were no differences in the expression MRGPRD in NP1 type 1 clusters, MRGPRD was one of the genes that came up as differentially expressed in the NP1 type 2. So these red cells here are cells with a higher expression of MRGPRD, and we can see that these are more pronounced in the high-fat diet. The same data can be seen and visualized by this violin plot as shown here. We can see that the differential expression of MRGPRD, even in NP1 type 2, is rather modest, but this increase selectively, uniquely in this subpopulation of DRG neuron, interestingly, has a functional relevance. And this is, I will be able to speak to these in the upcoming slide. So we had to validate the single cell result using, and we did that by using RNA scope on frozen section of DRG from our mice. And we can see that the expression of MRGPRD has here in DRG of regular mouse fed with the regular dye here in green. And now we are currently using a combination of different markers that are able to try to separate the two distinct subpopulations of non-peptidergic type 1 neurons that express MRGPRD in the high-fat diet. From the literature, we know that MRGPRD is a subpopulation of nociceptor neurons that are labeled by the voltage sodium channel NAD 1.8. And it's also that what we confirm on our RNA scope as shown here. We have RNA scope from DRG taken from both regular dye and high-fat dye. These are presented regular dye. And we can see that all the neurons that are marked with a probe against the messenger RNA for MRGPRD here in green are also labeled with NAD 1.8 as also represented in this graph. And from our single cell RNA sequence, we saw that at the transcriptional level, we found not detectable difference in the expression of NAD 1.8 within either the two MRGPRD expression clusters. We use again RNA scope to validate the different DRG subtypes that we identify with a single cell RNA sequence. And we can see, for example, again in DRG from a regular dye mouse, few examples that we use, for example, this probe against different MRGPRs that are known to be expressed in distinct DRG neurons. We also label with calc or other GPCR that are known to be expressed in distinct subpopulation of DRG neuron. We confirm what already described in the literature and what we found on our single cell data. We also utilize marker like, for example, TRPV1, and in particular here, when we see staining of messenger RNA for TRPV1 and MRGPRD, in particular on frozen section for regular dye and high-fat dye, we can see that in mouse, MRGPRD and TRPV1 are expressed in different DRG neuron subtypes, and that's true for mice control regular diet RD and mouse diabetic mice DRG fed with high-fat diet. Now, given the molecular difference in neuronal subtype between mouse and human DRGs, demonstrated by other laboratories already, it was important for us to validate our candidate target using human DRGs. And therefore, we validate our target on human DRGs provided by AnaBios. And here we can see RNA-scope experiment probing for MRGPRD and TRPV1 on the left on a mouse DRG, and on the right on the human DRG from a donor control. And then we can see that while in mouse, as already shown, MRGPRD and TRPV1 are expressed in two different subpopulations of DRG neuron, we can see that, on the contrary, in human DRG, as already demonstrated by other laboratories, first of all, we confirm the MRGPRD is expressed, but we can see that actually TRPV1 expressed almost in all the nociceptors, and it is co-expressed with MRGPRD, as this is different what we see in mouse DRG, so making perhaps the classic distinction between peptidergic and non-peptidergic DRG neuron proven in mice maybe less clear in human DRG neuron. It has also underlined the importance of validate our target using human DRG. We perform also RNA-scope on human DRG from a donor control, and also a donor with painful diabetic neuropathy. This is a human DRG. Again, we probe against MRGPRD and TRPV1. And we can see again MRGPRD is expressed also in a diabetic condition. And again, TRPV1 both in control and diabetic human DRG is more expressed in more DRG subtypes, and also co-localized with MRGPRD. Now, the single-cell RNA sequencing has demonstrated increased expression of MRGPRD uniquely in the subpopulation of the non-peptidergic type 1 and MP1 type 2 in the DRG neuron. But also we wanted to study the functional relevance of these changes in the MRGP expression in the alpha-die condition. And to do that, we perform in vivo calcium imaging, and that by conditionally expressed in the nociceptor NAV1.8 positive DRG neuron, the calcium indicator called GCampy6. This is a protein whose fluorescence intensity dramatically increased after binding intracellular calcium. And the image here shows the expression of GCampy6 in green in NAV positive DRG neuron. In collaboration with Dr. Richard Miller, we built a custom-made 2-photon imaging system where calcium signaling in lumbar DRG can be monitored in lamittomized patients, lamittomized mice, in response to manipulation of the paw, either with mechanical stimuli or with application of drugs. So to determine the changes, the functional relevance of these changes in MRGPRD expression in alpha-die condition, we use beta-alanine, which is a non-specific agonist for MRGPRD, and we inject beta-alanine in the paw of the mouse while recording calcium signaling in the DRG. And in the video here, we can see that DRG neuron, both in the regular diet control and also in the alpha diet condition, respond with calcium transient after the injection of beta-alanine in the paw. But a higher percentage of a DRG neuron respond in the alpha diet condition. The same can be visualized here, where we can see there is a statistically significant difference in the higher percentage of neuron responding to beta-alanine and injection in the paw in the alpha diet condition compared to the regular diet. This suggests that there is a possible an expansion of the MRGPRD expressing neuron in the alpha diet. Now, here we can see the calcium traces from the neuron responding to beta-alanine, And most of the responses, as visualized here, are starting within 200 seconds after the injection. And we can see that in general, we don't see a difference between the duration and an amplitude of the responses, which are quite similar when compared regular diet on the left and high-fat diet condition. But when we observe the high-fat diet conditions, we see that a population of neurons expressing MRGPRD, there were differences in a particular population in the nature of the responses. And this suggests that in addition to see a higher number of responding neurons, there may be a different sensitivity of those neurons in high-fat diet. First of all, only in the high-fat diet condition, we see a population of neurons expressing MRGPRD responding to beta-alanine injection in the periphery with calcium transient that have this oscillatory shape. Second, we can see that in a high-fat diet condition, a higher percentage of neurons responded with calcium transit that lasted longer. In the slide, slide pie shows this graph that the percentage of neurons where the duration of the response continues beyond 300 seconds is about 10% in the high-fat diet, in the regular diet here, but is double in the high-fat diet condition. Now we see that there were not differences in the area under the curve or in the maximum amplitude, but when we look at the number of peaks in responding neuron, we can capture this difference that exists in the oscillatory shape in our high-fat diet condition. And we can see these oscillatory shapes are very intriguing to us because there are characteristics of hyperexcitable neuron. So taken together with our single cell sequencing data, this reinforces our idea that maybe there are two distinct populations of DRG neuron expressing MRGPRD, and perhaps one subtype could be more important in mediating the hyperexcitability that we know to be so important in our model of PDN. Now, previously, we have shown that in the skin of the mice fed high-fat diet, there is this small fiber neuropathy, so degeneration of the axon. When we look at mice where all the nociceptor axons are genetically labeled in red, as shown here, we can see nice axon labeled in red, innervating the skin on a mouse fed high-fat diet, regular diet here. But on the contrary, in mice fed high-fat diet, there is a large depletion of this axon. But then to us it was a little bit surprising because we have seen that in our single cell, in our in vivo calcium experiment, we were able to see calcium transient when we we activate MRGPRD receptor in the periphery within the injection of the agonist beta-alanine in the paw. So if all these nerve, this fiber had degenerated, how can this signal be transmitted? And then what is the afferent that pick up this signal? And so we wanted to investigate the status of MRGPRD positive fiber in the skin of our model. And we did that by using this mouse where the MRGPRD positive fiber are labeling with the EGFP as the MRGPRD EGFP reported mouse. So we induce PDN in this model by either feeding a regular diet or a high-fat diet, and then we look at the skin in this model. These are low magnification section, and we can see the paw pad of mice. Here in the left, mice fed regular diet, and the right, mice fed high-fat diet. We can see that in general, as known, MRGPRD positive fiber extend to innervate here the outermost layer of the skin. But in these zoom images, we can see that MRGPRD positive fiber across the junction between the dermis and epidermis, here labeled with this white line, not only in the regular dye, but also in the hypha dye condition. So the skin of our model is different. There is some plasticity. There is some fiber MRGPRD positive that either remain and non-degenerate or even regenerate. And we think that potentially these remaining afferents expressing this excitatory MRGPRD receptor could be responsible for carrying mechanical odynia in this model. And in addition, given that these MRGPRD positive fibers are in the very superficial level, in the outermost layer of the skin, this could be a very interesting and accessible target for potentially an antagonist that could be even applied topically in the skin. So, for the summary of this first part or my talk, we have shown that using this single-cell RNA sequencing and comparing hypha dye DRG and regular dye DRG, we see that there is an increased expression of MRGPRD excitatory receptor, in particular in a subpopulation, uniquely in a subpopulation of non-peptidergic neuron in our model. In vivo calcium imaging also indicate that this is important as a functional relevance in the potentially could mediate hyperexcitability in this model. And also we saw that these fibers expressing MRGPRD persist even in the hypha dye condition. So on the translational perspective, we can say that MRGPRD is a very interesting target because it is a G-protein-coupled receptor, so highly druggable. It is an excitatory receptor with constitutive activity, so could potentially be activated and use excitability without ligand even. The expression is also known to induce excitability, response to mechanical stimuli, and also by being expressed in the outermost layer of the skin could be an interesting translational target for the development of new drug that could be disease modifying and also could be applied topically to treat our patients suffering with PDM. Also, I hope to convince you that actually single cell RNA sequencing combined with a validation on using RNA-scope both on DRG, mouse DRG and human DRG, and combined with functional experiment like in vivo calcium imaging can be a valid approach for identifying multiple targets that can also eventually help the development of disease-modifying therapy for our patients. So now, and indeed, we have used this method to identify other targets. So in addition to MRGPRD, this approach helps us to identify other few molecular targets. And these we are not discussing today. We have found that other molecules like the mitochondrial calcium important complex, so also some chemokine, including a chemokine receptor CXCR4, are important for increasing the excitability and then going through this molecular cascade of increasing calcium entry and lead to axonal degeneration. So now we are continuing to validate those targets in rigorous and preclinical study with a large number of mice, both male and female. We are also using a variety of pain behavior assays to test both provoke and spontaneous pains, which is very relevant. And we are using our hypha dye mouse model and also validating this target using human tissues. The good thing is that all those targets aim to reverse axonal degeneration. So we think that could help the development of a disease-modifying therapy. So therapy that they're not only treating neuropathic pain, but also able to restore the health of the fiber. And by reversing small fiber degeneration, underlying neuropathic pain in painful diabetic neuropathy and in other painful neuropathy, those therapies could potentially provide a long-lasting cure for our patients. Looking again with a translational perspective, an ideal therapy for PDN, painful diabetic neuropathy, and other painful neuropathy is one that could be applied topically. We know that topic application to treat neuropathic pain would be very appealing because it could bypass the side effects associated with a systemic intervention. So our laboratory now is very interesting to look at mechanisms triggering neuropathic pain in PDN in the skin. Now, the skin is a highly complex biological system, and a variety of neuronal subpopulation terminate here in the skin. We can see all these different subpopulation of fiber that terminate either in the epidermis or in the dermis. Of course, there are many other cell types that are found in the skin, and I just some few are represented here, and there are many others. And one of the main population that it is found in the skin are obviously keratinocytes. And keratinocytes are closely juxtaposed to epidermal nerve terminals. So it is possible that these provide an opportunity for crosstalk between keratinocytes and nerve afferents. We are thinking that this a better understanding of the communication between these afferents and keratinocytes can perhaps find mechanisms for boosting nerve regeneration, and therefore could develop therapeutic approach that not only restore cutaneous innervation, so disease-modifying approaches, but also approaches that could be applied topically. We have started looking at the bidirectional communication between keratinocytes and cutaneous afferents in PDN, initially in relation to wound healing, and mostly actually considering the effect that afferents have on keratinocytes. We know that in the diabetes skin, in addition to have small fiber neuropathy, so degeneration of epidermal fibers in the skin, it's characterized also by a slow regeneration of these epidermal fibers after wound healing. And there are many data, a study in the literature showing that this degeneration of these axons is also important for one of the characteristics, one of the major complications of diabetes, which is impaired wound healing. And indeed, we have shown that when we are able to restore the health of these epidermal fibers, we are already also able to improve and reverse this impaired wound healing in the high-fat dye mouse model. So it seems like there is a communication between fiber and afferent the skin and keratinocytes. And indeed, we know that afferents can communicate with keratinocytes by increasing their migration and proliferation during wound healing. Recently, though, many other labs have shown that keratinocytes have many roles than the one known thus far. So it is revealing potential additional role in keratinocytes in mediating a neuropathic pain. So now we wanted to better understand now this communication between keratinocytes and cutaneous afferents in painful diabetic neuropathy, but in particular considering the effect of cutaneous nerve terminal on keratinocytes, but also the opposite. So to better understand the potential contribution of keratinocyte to cutaneous denervation in PDN, in our laboratory, Dr. Abdelhak Belmadani has started to look at this communication between keratinocytes towards nerve fibers using these devices. These are microfluid co-culture systems where we can see, like in this graph, they have different reservoir. We have, for example, in this graph, we can see a view from the top of this reservoir in blue here, where we can grow DRG. We can grow, for example, we start growing adult mouse DRG in this reservoir. And in another reservoir, these two in red, we are growing adult mouse keratinocytes. And we can see that these specific devices also are connected by these microchannels as shown here. So these are particularly useful devices if we wanted to study the communication and the bidirectional communication between keratinocyte and cutaneous afferents. And so we culture using these multi-fluid devices, DRG neuron, as shown here. We have DRG neuron from adult mouse. In this particular case, we have mouse where all NAV 1.8 DRG neuron are labeled in red with the tomato. And again, we have these microchannels here, and in the other reservoir, we culture adult mouse keratinocyte. And using these devices, we were able to observe DRG neuron neurite outgrowth, as shown here, inside these microchannels, starting about within 7 days. So within 7 days, we can see the DRG neuron extend their axon with this neuronal trying within these microchannels. Interesting though, we can see that we can observe these neurite outgrowth in the microchannels from the DRG neuron only when the DRG neuron were in culture with keratinocytes. So when we had done, as previously shown, the DRG culture and keratinocyte, we did 7 days, we can see these axons standing to the keratinocyte. We also noticed that once these are crossing these microchannels, these axons were not able to go back in their previous compartment, but they are only able to continue to grow and indeed able to contact keratinocytes. However, when we grow DRG neuron alone in these micro devices, but without keratinocytes, we were not able to observe these neurital growth within these microchannels. So this indicates that indeed keratinocytes are secreting some factor, or somehow they are communicating with the DRG neuron. And so it is possible to make, therefore it is possible modulate eventually keratinocyte to regulate DRG neuron neurital growth. And eventually we potentially could think about target and therapy that could reverse axonal degeneration, underlying neuropathy pain in PDN and other painful neuropathy by modulating keratinocytes. So towards finding this possible target on keratinocytes, we focus on G protein caporeceptors expressing keratinocyte. Why? Because G protein caporeceptor are very highly druggable. Obviously, expressing keratinocyte could be accessible for modulation the activity of keratinocytes. And in our mouse model, we start to modulate G protein caporeceptor in keratinocyte using a chemogenetic approach. That indeed it is a G protein caporeceptor signaling platform using this designer receptor exclusively activated by designer drug or DREADs. DREADs indeed derive from molecular evolution from muscarinic receptor in order to bind a previously unrecognized small molecule that derive from clozapine and is called clozapine N oxide, CNO. And indeed, binding of CNO to this DREAD is able to activate all the classic G protein pathways. So there are many different kinds of DREADs, and there are one that are associated with the GI or GQ or GS, and we starting by expressing in basal keratinocyte that can be labeled with this marker K14. we start to express the dread that derive from muscarinic receptor 3 that is coupled with the GQ. So we know that binding of CNO to this dread will activate GQ on these basal keratinocytes if we selectively express this dread on these basal keratinocytes. So we can do that again using a genetic approach, and we selectively express G protein-coupled GQ with this M3 dread in basal keratinocyte K14 by crossing this mouse, the K14 Cre mouse with a Cre sensitive dread mouse, M3 GQ dread flox that is being generated in the laboratory of Patricia Jensen. And so when we cross these two mice, we obtain mice where all the K14 basal keratinocytes are now expressing these N3 dread coupled with GQ. So we could potentially then activate this GQ in keratinocytes and see the effect of this activation in keratinocytes in our model. And that first, though, we had to demonstrate that indeed we were able to express these dread on basal keratinocytes. And we did that by confocal imaging analysis. And we can see that in this confocal macrograph, we can see that in the skin on mice where we express this GQ dread, this GQ as M cherry, so we can visualize the expression of these N3 GQ dread here in red in the basal keratinocyte. And we know that these are basal keratinocytes because as shown here in green, these cells are also stained with an antibody against this marker K14, then identify these basal keratinocytes. And we can see in this co-labeling, indeed, we are able to express this dread GQ in keratinocytes. So then the question is, what happens if we manipulate these GQ on keratinocytes in the model, in the skin? So in order to activate these dread, we have to inject CNO. So we had now this protocol in our mice where these GQ are selectively expressed in basal keratinocyte. We inject either saline or CNO 3 times a day for 4 days. And then on the 4th day, we harvest the skin of these mice. And then we look at the histological characteristic of these mice, or the skin of these mice, we can see very interesting finding. And one is that in the skin of mice injected with CNO, there is a thickening of the epidermis. So we can see that also quantify here in mice injected with CNO, where is a statistically significant increase of the thickness of the epidermis compare mice just fed, injected with vehicle. And we did with the tracing, we can see that this is much thicker. In addition, we also saw that in epidermis of mice where we inject CNO, the keratinocytes increase their proliferation. And this was done by BRDU incorporation. So here we can see that in mice injected with CNO, there is these keratinocytes in the epidermis there is statistically significant increase of proliferation of this keratinocyte compared mice injected with vehicle. Also, we noticed that we also noticed increase in the keratinocyte migration be assessed with the BRDU pulse chase. And we can see in these images, as also we can see quantify in this graph here, there were the migration of keratinocytes in mice injected with CNO. It is much higher, so they migrate much more in the mice injected with CNO compared to vehicle. So we see a lot of change in the keratinocyte, but also we modulate GQ within the keratinocyte. So but our question is, was possible to modulate keratinocyte to see some effect on the cutaneous effect? So then we look at what happened at innervation in the skin in this model. And we can see that actually activation of these basal keratinocyte, activation of GQ in basal keratinocyte induces loss of innervation in the epidermis. And that can be, we can see here, where now these fibers are labeled with this fiber marker, the PGP 9.5 in green. And we can see that in the skin of mice infused with saline, we have nicely this fiber innervating the epidermis as a normal innervated epidermis, as shown here in gray. However, in the skin of mice where we activate GQ in the keratinocytes, so injected with CNO, we see not only again a thickened epidermis, but also a reduction of these intraepidermal fibers. So there is a degeneration of this axon, and this is very much similar to what we see in the skin of mice fed high-fat diet. So in diabetic mice, as well as in patients, we have this degeneration of this axon, and then we could see that is also quantified here. We have a reduction in innervation in the epidermis. Other things are occurring in the skin, but focus on the epidermis, we can see there is a reduction of the innervation in epidermis. So you could imagine we could modulate GQ in keratinocyte, for example, blocking them with an antagonist and then stopping this axonal degeneration. So this is an interesting hypothesis, and then obviously these mechanisms are very complex. We have both systems, they are very distinct by different subpopulations. So we have keratinocytes with different kind of population in the skin. We have then the different subpopulations. So we decided that in order to try to better understand this phenomenon, we had to use, again, a single cell transcriptomic approach. So in this case, we decided to use these mice where, again, the GQ thread are expressed selective in K14 basal keratinocyte. We activate these GQ expressed in basal keratinocyte with CNO as previously. So we use the same protocol, injecting either saline or CNO 3 times a day, and then this time though, on day 5, we harvest skin and lumbar TRG. These are very new experiments. So we did obtain a viable single cell suspension from both DRG, lumbar DRG, and epidermis in the skin. And both in mice that are female and male mice. And today I will present only data from male and single cell RNA sequencing of DRG neuron. So again, we are able to obtain a valuable suspension for DRG, lumbar DRG, and we use again the 10x platform for single cell capturing and barcoding. And then we did our comparative analysis. And again, we were able to identify cell types as well as conserve marker in the 2 conditions. We're able to separate both a non-neuronal and neuronal clusters, like we already done and also previously described in the literature. Again, the analysis is still ongoing, but our comparative analysis, we can see a big difference when we compare mice injected with saline and or CNO. So indeed, activation of GQ in keratinocyte resulted in identification of we can see different subpopulation, but we can see that in the population on the DRG and mice injected with CNO, we have a new cluster, a new cluster that we are not able to identify in mice injected with saline. And so this is something that indicated that indeed modulation of GPCR in keratinocytes is able to change the gene expression profile of DRG, and then could have a due impact in DRG neuron plasticity. So I tend to summarize now the second part of this talk. I hope again I convince you that today the really single cell RNA sequencing of peripheral somatosensory neuron can be a good strategy in helping us to discover target for painful diabetic neuropathy. This target, by targeting axonal degeneration, could be helping the development of disease-modifying therapy, and also could help the development of therapeutic that can be applied topically. If this mechanism, this target could be, for example, found in the skin by better understanding the communication between skin and cutaneous substrates. And obviously, I would like to thank my team that in this translation approach included my clinical team. This is the Peripheral Neuropathy Clinic and the Charcot-Marie-Tooth Association Center of Excellence. This is the people helping in the clinic. And of course, I'd like to thank the team in my laboratory. And that obviously perform all this work, and I like it for all the work and for their support. In particular, I like to thank Nirupa and Dale that perform majority of the experiment that and the lead majority of the project present today. I would like to thank Danjen for her excellent technical support, and Abdel that is leading this exciting project of skin-nerve communication. We are looking for postdocs, so that postdoc position open. So please contact us if you are interested to join our team. And I would like to obviously thank our collaborators. I would like to thank AnaBios for providing human DRG from donors. And I would like to thank all our funding agencies. And I would like to thank you for participating in this webinar. And also, if you are additional updated, I would like to invite you to this Peripheral Nerve Society meeting or Neuropathic Pain Consortium meeting that we occur in May, 14-17 May, in person in Miami. So after this, I'm happy to take any questions.
Thank you, Daniela, for such an excellent presentation. Very interesting. We have a lot of questions already. So please, if you look at the GoToWebinar panel, you can write your questions in or you can raise your hand. We will likely go past the top of the hour, so hang in there. There's some really good questions, and we'll do our best to get to all of them. If you do have to leave to another meeting, be sure to download one of the brochures in our handout section. So I'll start with a quick question as people start to write in more. Daniela, the microfluidic co-culture chamber, is that commercially available or is that something that your lab developed?
No, this is commercially available, and yes, these are commercially available. There are actually very, very interesting devices to study communication between cells. We are using them where we can culture not only mouse DRG, but eventually human, both DRG and keratinocytes. So yeah, this is actually available, though they need a little obviously troubleshooting about how to grow these cells on this, and even to allow the growth of these neurites within the microchannels. There are different sizes of the microchannels as you adapt to the different culture. But yes, they are available.
And which company makes that?
Is Xena, I think. I will provide this information. I think Abdel knows for sure.
Okay,
If
I'll
Abdel
check
is
later.
on the call, please, if you are on this webinar, Abdel, unmute yourself and please provide this information.
Yeah, let me unmute him. Let's see here.
Sorry about that.
It's okay. So here's a question. Are there any RNA expression differences among the DRGs which are from different locations, such as lumbar versus cervical?
Yeah, that is a very interesting question. And actually, there are different, obviously, already in the literature, different single cell RNA sequencing approaches that have been done and in different publications. Either other author, some colleagues have used all the DRG from cervical to lumbar, or also only lumbar. We focus on the lumbar DRG because we are focusing on the degeneration that occur in the paw of the mouse. So the dermatomal distribution basically indicate that there are the neuron that extend their axon over there. So we are focused on that. We have never done a comparison, and as far as I know, there is not really published a comparison on the gene expression on this level. But we didn't do that.
Okay. And you mentioned in the second part that you focused on male rats. What about the first part? Was that male and female rodents?
Yeah, so we are now, I mean, this single cell experiment now they are becoming more and more available in a sense. So initially, now we do more duplicate and we use mouse and female and male. We initially did all the experiment male, but now we are doing everything male and female because obviously, I mean, the other laboratory has demonstrated huge differences when compare male and female, especially in all mechanisms underlying neuropathic pain. So the gene expression profile has to be done. We did not compare them yet, so we had to analyze all this data, both in the DRG from mice with this dread in it, activated, non-activated, and also high-fat diet, regular diet. So we still have all these data from single cell RNA sequencing where we compare mice, male and female mice in our models. But this is absolutely something that needs to be done. And there are already known differences that other laboratories have published already, of course.
Yeah. I think this question relates to the first part of your presentation. Have you measured calcium uptake?
Calcium in the DRG?
Yes, I believe so.
Yeah, so I mean, in the part of the calcium influence, there are so many other cellular mechanisms that we are trying to better investigate, in particular related to the mitochondria calcium reporter. We are now better understanding, first of all, the concentration of calcium intracellularly and also in the mitochondria. We are trying to apply the GCAM-C6, this calcium indicator targeted to mitochondria. So we are trying to see exactly how much is the calcium entry into the mitochondria and the calcium, where the calcium comes from, if from extracellular or from the storage, calcium storage, all these kind of more electrophysiological calcium question that we are doing in our calcium projects, like the MCU calcium project.
Right, plenty of work to do, indeed. Someone asked, have you seen any excessive scratching in your diabetic animals?
In assessing scratching just in the high-fat diet, no.
Okay.
And I know this is obviously a very important question for the role of RGPRD in itching, but we have looked actually by video. This is a chronic condition, so it's a little bit difficult to monitor, but for what we know, for what we see in our video, we did not see increase of itching and scratching.
You know, it's interesting. I've been to a presentation on fatty liver disease where they use, I don't know if it's the same high-fat diet, but when they take some of the rodents off the high-fat diet, there can be reversibility. Is there any reversibility if you do that in this case?
This is a very, very, very interesting point, which actually has been shown in a patient. So a study Dr. Gordon Smith and others have done studying in patient with metabolic syndrome, and they have checked the skin. So they found this small fiber neuropathy in the skin, and then they apply exercise, a diet modification, and that prove a reversal. So this small fiber neuropathy can be reversed by removing that diet. That's also what happened in our model, but in patient too. That's possible. So that could be, in reality, the most effective disease-modifying therapy.
Right.
that it is already shown, has been shown in few groups now, patients.
Here's another question. You show that NAV 1.8 positive fibers completely disappear in PDN mice, but MRG, PRD plus fibers persist.
Yes.
Which NAV 1.5 plus subset do you think are degenerating then? And are the MRG, PRD plus fibers that persist no longer expressing NAV 1.8? And if so, how do you reconcile these two findings?
Yeah, so this is a very interesting question, puzzling. So actually, this is just an hypothesis, but our idea is that these fibers perhaps are more regenerating fibers rather than remaining fibers. So it perhaps talks about more plasticity in the skin of these mice, where when there is a degeneration, other things happen. And so perhaps these are more premature, more de-differentiated DRG neuron, this fiber express MRGPRD receptor, perhaps their calcium waves is still immature. I mean, it's possible that talked about more the plasticity of this fiber. So the skin and effect diet is completely different. So what happened in the DRG is not only degeneration, but regeneration, and perhaps a different stage of differentiation in these DRG neurons. So this is what we are trying to investigate with the genetic tools to ask these questions.
Okay, thanks. Someone asked about the human DRGs. How many DRG samples did you use for the human?
For this one are very few. We only obtain from AnaBios 2 for each, and so these are very, very thing. We are obviously we have to have a system or provide a much larger group of DRG. Right. So these data I present today is because they've already shown, already known that in human DRG 3.1 was extended, but to really make a conclusion of that, and in particular, we would like to look at the population of patients with PDN, so donor with PDN. Is obviously that's much more difficult, in particular if we aim to use DRG from donor clinically characterized, where the neuropathy, the PDN is very clinically characterized. That's a much more challenging thing to do.
And that person asked which lumbar segment for those?
L4.
L4.
For human, L4, yeah. From AnaBios.
Yeah, and the person was asking how they were obtained, and I can answer that question. So AnaBios has a very extensive network in the US only of hospitals and transplant centers. And so we obtain DRG sets from T1 down to S1. And so we use proprietary solutions to get them to our laboratory in San Diego very quickly to minimize ischemia. And the fact that we can do physiology experiments attests to the high quality of these, and very, very minimal RNA degradation. We have very high RIN scores, close to 9. So I think that's why you were able to get those successful experiments done, Daniela. Mm-hmm. Here's a lot of questions. We'll do our best to get to all of them. Wondering if any nerve conduction data to support the innervation with CNO?
No, we didn't do that, the conduction. And this is an interesting question, looking more at different kind of fibers that are more the large fibers. So we focus more on the small fibers that are expressed in the intraepidermal, in the epidermis. As different capture this fiber with electrophysiological nerve conduction study. So all these questions on electrical nerve conduction study in these mice would be very interesting to address looking at this large, more myelinated, either sensory or motor fiber, where you can be visualized either the conduction velocity or the power of the motor compound motor potential. So all these parameters are very interesting. We are not focusing much on this large fiber. We focus more on the small fiber degeneration, which is captured by histological finding, microneurography, but not too much about nerve conduction studies, EMG.
Okay. Is there any evidence that MRG is the driver for excitability in DPN, or just a marker for fiber types that are otherwise more excitable in DPN?
Actually, this is a very interesting question, and that's the reason why we really wanted to have this in vivo calcium proof that peripheral activation of these excitatory MRGPRD in the pore, on the skin, so by injecting beta-alanine, was able to induce the calcium transients in the DRG, the lumbar DRG, in vivo, in mice that are anesthetized. And so basically, that was the main question in there. Is like, okay, is the MRGPD just expressed or is it functional relevant? And indeed, the finding of the calcium are interesting, especially in the two type of calcium waves. One oscillatory shape, not only in expansion of the number of DRG expressing MRGPRD, though responding to beta-alanine. So this is a very crucial question. So we think because we found these in the vivo calcium, indeed, MRGPRD expression has a functional relevance in modulating the excitability in this particular subtype of the DRG neuron in our model.
Very good, thanks. The thickening of the skin with CNO and HM3 DQ due to keratinocyte division and migration may have important implications in wound healing in diabetes. Have you tried this model in a wound healing model?
That's a very important question. And we are collaborating with the chairman of dermatology here at Northwestern, Dr. Amy Pella. She is actually expert in wound healing, impaired wound healing in diabetes. She's a dermatologist, of course, and so we are working with her to answer this question because, again, impaired wound healing is a very important complication of diabetes, leading to amputations. It is a very, together with the neuropathy, it is a really bad combination, impaired wound healing and degeneration of the fiber, where you lose sensitivity to. So this is a very important question, and that's why this model, we will ask both questions, but one in particular, Dr. Amy Peller, that is expert in wound healing.
Okay. Does keratinocyte activation using DREADS lead to an immune response in the skin? And if so, does the increased thickening lead to inflammatory pain?
Yeah, that's a very important question, and we are looking at that's why actually we decided to do single cell RNA sequencing on the epidermis, because again, this is the skin is a very complex system. So it is not only keratinocyte fiber, a lot of stuff happening, and there are inflammatory infiltrated, and we know that the thickening of the epidermis is also characteristic of other dermatological conditions. I'm learning from collaboration with Dr. Amy Peller, a lot of these dermatological condition and relation with inflammatory markers and stuff. So of course, it's much more than only keratinocytes and fibers. And hopefully from our single cell, we were able to pick up some of those changes as well.
Very good, thanks. Here's a question from Kate Sadler, who I think is still in the audience. She's wondering, did you perform any behavioral tests in the DREAD mice following CNO injection? She's wondering if this manipulation would change sensitivity of mechanical or thermal stimulus application.
Yeah, that's a very important, very interesting question. And we are now in this preclinical study, we have to use large number of mice, and male and female. We are now really applying different behavioral assays, not just focusing only on the evoked, but also spontaneous. So these are ongoing experiments. Obviously, these questions are crucial because if we wanted to target also the neuropathy pain, only the axonal degeneration, we have to assay all these. These are a very important questions, still ongoing.
Very good. We'll just do a couple more questions. How do you identify neuronal subtypes in the single cell RNA sequencing?
See, that's actually a work that has been done, and so we are much facilitated now that many other laboratories had performed already in the last 10 years, single cell on DRG neurons. So now we know that the DRG neuron are subdivided different subpopulations, and we know the markers. We know the combination of molecular markers that identify specific subpopulation. So we can use those, the precious informations, and apply when we do our clustering in our single cell analysis. So in the skin, it's still a little bit more complex because again, all these markers, we have to do more work in kind of finding all these markers that identify the different subpopulations. But in the DRG neuron, in mice, and even now in human, a lot of work has been done already from other laboratories.
Very good. And I did notice that Abdel wrote that Zona Microfluidics is the company that makes the co-culture chambers.
Oh, wonderful. Thank you.
Yeah, I'd like to try that at AnaBios with our human spinal cord slices connecting to DRGs. That could be very interesting. So yeah, we didn't get to all the questions. Gary, if you could take a snapshot of the questions, we can send those to Daniela and perhaps she can follow up with the audience. So obviously it's a very interesting presentation, a lot of questions, very much interest. Thank you so much for taking the time. We look forward to the publications that are coming out, and thank you for giving us a preview of that. And I'd like to thank Gary Watkins for putting this together. Thanks for everyone for attending. And stay tuned for the next AnaBios webinar, which will hopefully be next month. So thanks everyone for attending, and we'll see you in the future. Thanks, Daniela.
Thank you. Thank you. Bye-bye.
Bye.